have Dr. Robyn Barbato. She is a research scientist at CREL, which is the Cold Regions Research and Engineering Lab in Hanover, New Hampshire. Her specialty is microbiology and permafrost and other environments. And today she is presenting on the permafrost microbiome and looking at past diversity from the late Pleistocene up through the Holocene. So with that, if everyone is ready, I will turn it over to Robyn.
Thanks, Andrew. Am I okay on volume?
Sounds good to me.
Okay, great. And thank you, Andrew and IARPC for inviting me today. So as Andrew mentioned, I'm from the Cold Regions Research and Engineering Laboratory stationed in Hanover, New Hampshire. And we have our sister lab in Fairbanks, Alaska, where Andrew is stationed. Today, I'm excited to talk about the invisible part of permafrost and how it holds the key to a wealth of information hidden inside. This work is in collaboration with researchers from the University of California, Santa Cruz, and the Fort Wainwright Archaeology Program in Fort Wainwright near Fairbanks, Alaska. And once again, I'd just like to thank the Interagency Arctic Research Policy Committee for the opportunity to share our findings. So the landscape presents a level of heterogeneity between vegetation, topography, and hydrology at the site scale. And as we zoom into the soil pit, we can see the active layer of soil and the permafrost beneath. Soil particles and micropores are heterogeneously distributed within the permafrost core. And here's a small depiction of a soil pore. And there, in cartoon form, is some green organic matter, the particles themselves, including the clay particles, which have really great binding capacity, some bacteria and fungal hyphae, and then water, whether it's in a liquid or solid state. So we are testing whether we observe differences in microbial community composition from these layers within permafrost because of differences in ecosystem traits at the time the feature formed. So we use molecular techniques such as DNA extraction and sequencing to capture the abundance of bacteria, archaea, and fungi present in the permafrost. So these microbes are not static, but rather are interacting with one another in mutualistic, symbiotic, antagonistic, competitive ways, adding an even greater level of complexity to this problem. So this could make your head explode, but we need to put our craniums back on and do the best we can to observe these interactions given the tools that we have. And there are different tools out there, and the one that I'm going to talk about today through DNA extraction is unfortunately just a snapshot. But in that snapshot of a video, imagine, you can actually capture a lot of information. And we combine this with other methods like microscopy. So we visualize this complexity, see the large boulder-like soil particles and tendrils of fungal hyphae connecting these particles together. So this is through scanning electron microscopy. Just to add this information so we remember that we're working at the soil scale and not just a bunch of A, T, G's, and C's on our computer screen. So as most of you know, I'm sure way more than me, that permafrost comprises a large area of the globe. Here in shades of purple are the different flavors of permafrost, ranging from isolated to continuous. Our study areas focused in interior Alaska near Fairbanks, where permafrost is indeed discontinuous. It's projected to change dramatically as temperatures rise to about 5 degrees C by the year 2100. And permafrost and other ancient soils, termed paleosols, harbor this trapped biochemical evidence of the environment at the time the feature was created. So the nutrients, mainly carbon and nitrogen, drive the organisms present. And these research questions, I at least feel, tie into IARPC's research plan on field-based empirical data acquisition to understand relationships between ecosystem processes and permafrost processes. A lot of effort is going into what happens during thaw, but this one here is focused on simply taking those features and understanding what's there right now. And we have limited resources to understand that. We have fossils, we have DNA, we have isotopes, but we're all just trying to come together to create this story with, quite frankly, some limited resources. So let's take a stroll through time and consider what major events occurred. Now, I thought this was pretty straightforward, and I'm realizing it's not, and a really fascinating kind of a documentary where Eski Willerslev is featured really highlighted the back and forth nature of what happened in the past. So here's our timeline. The Holocene, starting from modern time on the right, and then the Holocene as we proceed left about 11,500 years ago, and then the late Pleistocene where we stop at 60,000 years, but obviously time occurred before then, and I consider this ancient biodiversity, but my colleagues in paleoecology go back even farther, so take that with a grain of salt. So about 55 to 21,000 years before the present, megafauna roamed, mammoth, woolly rhinoceros, I didn't even think that existed, bison and horse roamed with the dry tundra and forbs. Then about 24,500 years ago was the last glacial maximum, where there was a cool, dry climate. Note that interior Alaska was not glaciated at this time, which is a common misconception. Then there was glacial retreat, where the glacier started retreating around 17,500 years ago, and there was more moist tundra environment. A mass extinction, this sad mammoth here, at about 13,000 years ago, where 3/4 of the large mammals died. And this graph shows the temperature differential between the Pleistocene and the Holocene, so these large temperature swings by proxies of stable isotopes, and then the Holocene being much more stable. And then this is just a quick snapshot of the Holocene as we move from herbaceous tundra to shrub birch tundra, and more towards the boreal forest with sporadic tundra or barren that we see today. So microbes drive these fundamental processes in soil. So one fundamental question is, what were they doing while all of this was happening above the ground? So this schematic shows our sample locations and how those samples represented different time periods, from modern times to the Holocene to the late Pleistocene. We're wearing the bunny suits, not for fashion, but so that we don't contaminate the samples that we're collecting. However, they proved to be quite warm when we were hiking in the snow, so they have dual use, I should say. And now what I've done is I've flipped our axis vertical, and on the top is the modern times, and on the bottom is that 11,500 years before the present. So our study objective is to examine the relationship between biochemical signatures and evidence of past ecosystems to understand the basic properties of how biodiversity was shaped by these broad global changes. And then we can use the past to better forecast future changes. So this image represents a bluff in interior Alaska with paleosols ranging from about 340 to about 7,000 years before the present, with 2 tephra in between. And just recall that the strategy that we take is that we sample these soils, extract DNA, and try to understand broad changes in microbial diversity through sequencing. So on this ordination diagram, each point represents a soil sample and the community assemblage within it. So our findings show that the bacteria and archaea, and then we have the fungi, that their sequences obtained from the modern soils do not reflect what we collected in the Paleosols. So on this, in this sense, that there's something different that's been happening in the past as compared to what we're observing in the current times. And you can see a broad kind of I guess a vast change even within the modern soil, suggesting that they're heterogeneity, but they're not even close to the Paleosols, particularly on the first ordination axis, which captures the most diversity of the system. So another thing that we did was, the first question is, well, what main soil properties drive this? Because a lot of people have done really great research tying changes in microbial diversity to pH or moisture or organic carbon, which are drivers of the community. And when I ran this in a canonical correlation analysis, none of those properties were driving any of these community clustering. So that was quite interesting, suggesting that there are alternate drivers. So let's zoom in and try to look at the bacteria a little bit more and show their relative abundance. So these are the dominant bacterial phyla present in these samples. On the x-axis is their relative abundance. On the y-axis are these different soil samples from modern soils all the way down to 7,000 years before the present. So those are Paleosols 2, 3, 4, 5, 6, and 7. I don't know what happened to 1, so you can save that question. It's an unknown to me as well. And below highlights the dominant traits, because I think a lot of times, particularly for non-microbiologists, when we talk about this organism does this organism does that, you can't see it. It's hard to imagine. So these are some nice pictures of what the representatives look like, if you will, and just broad things that they can do. Because one way that we use this information is to capture who's there so we can figure out what they're doing. And there's other techniques to really hone in on what they're capable of, but this is one way. So first things is that these are also with a significance of less than 0.05. So this is where we've seen significant difference. Other phyla didn't exhibit this. And they don't exhibit the same shape, which is the first thing that I considered, that their relative abundance is changing through time in these soils. No proteobacteria, which is oligotrophic, commonly found in soil, has broad metabolic diversity. And another soil organism, Verruca microbial, they are really abundant in modern soils, but that drops off significantly as we go back in time. Another interesting thing about this diagram are the chloroflexi and nitrospirae. And so these organisms, like chloroflexi, are known carbon fixers, nitrospirae are known nitrifiers, and after approximately 4,000 years before the present, they become quite abundant, and then and that wasn't the case in modern soil. So this suggests broad differences in ecosystem function. Perhaps there was a more aquatic environment where photosynthesis was more common, and that's why we're seeing that signature. We also did the same for fungi because they are part of the ecosystem, though often ignored. Usually people only have hyper focus on the bacteria, but they are also really important in soil function. And these are the classes of dominant fungi. So the first 5 are Ascomycota, and the last 3 are Basidiomycota. Basidiomycota are akin to mushrooms, are within that clade. And I think the largest organism by biomass on the planet is a fungus somewhere in England. So, I mean, they're really extensive and can have a broad reach, if you will. So you imagine those little hyphae extending out. So the patterns of the Archaeorhizomycetes, Agraricomycetes, Microbiotriomycetes and Poussiniomycetes are really striking at this 340 years before the present. So they have this significant increase and then drop off at this time. And this could be related to cellulose, decomposition, and what we're doing now is relating this back to what we find in the literature regarding the vegetation patterns to see, as I showed earlier, that there were different changes in vegetation. So can we couple that to what are, I would argue, the foundational part of the ecosystem being the bacteria and fungi. So our metagenomic data was really interesting because it highlighted major changes in function with an increase in the traits in blue and a decrease in the traits in red in these older soils. So these organisms have a higher stress response in the past, higher aromatic degradation, something that a lot of people care about, but particularly the Army, are different energetics and also petroleum hydrocarbons that are contaminated, have contaminated lands in this region. So that's of importance. Another thing is the increase in virulence is really curious because one concern is that as these ecosystems change, permafrost thaws, that there'll be more propensity for disease. And I think one classic example was in Russia where a caribou that was infected with Bacillus anthracis, so anthrax, emerged because of the permafrost thaw, and I think a few people died from that. So it is a relevant topic to explore. So let's go back even farther into the late Pleistocene, and I'd like to share the Urtick permafrost tunnel. Now my geophysicist colleagues, particularly in Alaska, will cringe as I say this. I've had a lot of really interesting discussions with them, but as you walk into it, it's almost like you're going through time in the time frame that we're interested in, at least. So it's really helpful to have a resource such as this at our fingertips, because if we were to take a vertical sample, we're in an area of discontinuous permafrost, we don't know if we'd hit permafrost, we have no idea what age it is, and this tunnel in particular is really great because there's a lot of information on it, and then there's also chronological data sets being generated, which is helpful to us. There's another tunnel that's almost complete being built. I guess that could be up for debate too, but you can walk into it. And unfortunately, that doesn't have the range of time. So it shows that this one's quite interesting. And I think it was the Sochi Olympics a few years ago where Russia's permafrost tunnel, they used it as an attraction, which made me cry a little bit inside because it's a great research resource. And they were using it more for entertainment, but I'm happy that this is limited to tours and research, and we don't have parties in here, so as not to contaminate what we're looking for. So we collected permafrost cores at specific points in the tunnel, representing the about 15,000 years, from 20,000 to 35 years before the present. And from a domain level, we observed really broad shifts in biodiversity. And that is exemplified here, but also what's really interesting is particularly at 45 meters within the tunnel, there's incredible heterogeneity. And when we looked at the soil properties, which again is really important to combine with microbiological research, because that's what might be driving the system. This sample had a totally different texture, and we have visuals to show how different it is. So naturally, it's not surprising that the community would be so different. And again, this plot is showing that samples that are closer together, those communities are more similar, and then ones that are farther apart are more different. And the x-axis is PCOA1, and that captures the most variation in the system, so that's important. The other thing to note is that bacteria dominated our samples. So this is not surprising, but also when you walk into the tunnel, there's bison horn, there's mammoth femur, there's a lot of physical evidence of other organisms. Granted, their DNA is exposed and could be highly degraded over this time, but it just shows, you know, if you're looking for eukarya, you have a harder time finding it perhaps than bacteria. Then bacteria are not your friend, whereas usually for us they are. So this is important and important for our colleagues. When we look just at the bacterial phyla, we see separation for the different areas within the tunnel, and we see a different set of dominant organisms. And these are proteobacteria, actinobacteria, and firmicutes. So firmicutes are known spore formers. That makes a lot of sense because as this environment is sitting at -2 degrees C, so it's quite cold. I calculated it's 491 days for these organisms to respire 1 gram of carbon. So that's a long time, so they're really slow going. But you can see that they're different than the paleosols, and also of interest are these actinobacteria, which are notorious for creating antimicrobials, antifungals, and they have produced these really interesting bioactive metabolites that we're looking more into. So in the same sense of, you imagine these megafauna roaming the area there, and you have to be careful with sequencing, because if I see alligator DNA in the data set, probably didn't occur at that time. So that's I would say a limitation. You really have to understand the system and combine it with other data. But our metagenomics showed the decrease in polysaccharide utilization, carbon fixation, and cold shock proteins, whereas quorum sensing, plant alkaloids, and biofilm formation increased. And this is it's very different from those exhibited in the paleosols, and also important for different kind of materials that people are interested in harvesting. So I like to think of permafrost, or express it as the Amazon of the Arctic. So imagine this wealth of potential from the microbial community that we have yet to scratch the surface. So in conclusion, and this is a beautiful picture from Udavik, which we've done some other field work in, that microbes are the dominant source of DNA in these soils. The ancient climate, flora and fauna changed the microbiome in ways that the modern soils did not. Despite these drastic climate changes, there is this core microbiome, and suggesting a potential for these really interesting materials and products that they make. And because the proportion of organisms reflected more of substrate micronutrient utilization rather than weather-related conditions, we feel that those influence community composition in these soils, and actually are the root of that ecosystem function, which we can combine with our colleagues who are looking at pollen and different vegetation that may have existed. So I'd like to thank the Assistant Secretary of the Army, the Acquisition, Logistics, and Technology, for our research, in the Army for appreciating having a full understanding of permafrost microbiome and processes that have occurred in the past and may emerge in the future. And Fort Wainwright Directorate of Works Archaeology Program was instrumental in our access and understanding of the samples that we collected. Thank you for your time.
Great, thanks, Robyn. We have time for some questions. Does anyone want to bring up anything from the talk?
If it's okay, I can start. It's Vladimir here. Can you hear me?
Yep,
Yes.
I can hear you.
So maybe I missed it, but so you're just seeing DNA, but you don't know if these organisms are alive or dead. It's just so you cannot tell, right?
Right, yeah, so we've tried extracting RNA, which would indicate an active community, but we're more interested in the DNA materials as as fossils, if you will, of what occurred. We do have a sense of the proportion of the community that's active based on other techniques such as respiration and different staining techniques that we use for at least bacteria. But yes, this is DNA. This is just what's there.
Okay. And the second question, kind of related. So how mobile the microorganisms are? They can move through the layers, or they prefer not to?
So I think that's a fabulous question, and I've had a lot of conversations about it. And I think I want to argue that we just don't know yet. I think there's been so much research on the geophysics, the geochemistry of permafrost, and while there's been a lot of really great research on the permafrost microbiome, these sorts of questions are left unanswered because there is a fluid nature. I mean, think about ice wedges forming, or even within the thinking of it as a fluid itself and whether they can move. I would argue not very far, and it depends on the porosity and interconnectivity, but I have not seen a study that actually tested that directly.
Thank you.
If I could pipe in here, we have some data from the Fox Tunnel, some metagenomic data that suggests that the older samples have at least a higher genetic potential for motility. So increases in chemotaxis genes.
What does it mean in a normal language, please?
So the genes that encode the proteins that drive the motility machinery, so that drive the flagella, that can sense environmental stimuli that will move the cell towards whatever stimulus. We find that the genes encoding the proteins for those systems are more abundant in the older permafrost in the tunnel.
But I at least haven't seen anything, and maybe you have, Rachel, where people actually tested that in situ. I haven't seen it. So I think there's evidence of proteins, genes, but I think going back to Vladimir's question, we're hypothesizing based on some evidence, but there's nothing that's shown that fluidity or even combined it with some interesting soil physics.
Yeah, I know that in Jenny Holtman's paper, they found more proteins related to motility in permafrost than in the active layer or bog. So not talking about age gradient, but the presence of the proteins. So that does indicate that there's some motility.
And I think that would have broad implications for what we're trying to do using DNA, because if there is evidence that they're modal, then what's the fluidity between the age ranges that we're trying to capture? So yeah, I think great question. I think more research is needed.
Thank you.
Hi, it's Martin Jeffries. Vladimir just drew attention to the fact that biologists use a rather different language to physical scientists, of which I am one. And so I've heard a lot of terms and phrases and words that don't mean an awful lot to me. And I've never taken a biology course of any kind in my entire life, so I'm particularly handicapped. So I'm curious, what for example, does quorum sensing mean?
Yes, so I apologize up front. I wasn't sure the level of detail that I should go into, and obviously I fell into that trap. But quorum sensing is a way for organisms to, through chemicals, sense other organisms or other processes. So think of them as communication molecules.
So pheromones would fall under that category. Would that be correct?
I think so. I consider that more for larger organisms, but yeah, I think that's a great analogy.
Okay, thanks.
And I'd like to talk with you, Marin, too, if you're interested in provide it more in terms where we can relate.
Okay.
I'm curious the extent to which reconstructing communities like this has been linked with either plant communities or at least collections of functional types. Is there a lot of ongoing work right now doing that throughout Alaska, or is that something where there's a continuing opportunity?
I would argue a continuing opportunity. I think folks are doing that. They're taking these shotgun sequencing approaches to understand all the DNA that they can get in the sample. I've seen connections between some pollen reconstruction and some megafauna, but I think we're just scratching the surface, to be honest with you. Sometimes just provided whatever funding that you have and understand, you know, your research question, we become quite hyper-focused, and it takes so much energy to understand your one area of interest. But I think, yeah, there's more of that occurring, but we need a lot more. And the other thing is extending the breadth of our understanding. Through you, Andrew, not all permafrost is created equal, is what I've learned. And so how do we connect more to get a broad sense? You know, we're really utilizing, I know of a lot of groups utilizing the Krell permafrost tunnel, but that's one. I mean, right next door, are we seeing similar communities? Can we disprove, you know, the things that I've said just based on that variability? And being a field soil microbiologist in the non-frozen state and understanding the heterogeneity, I wonder, but I don't have, I haven't yet tried to look at 2 points spatially separate, but representing similar ages to see if they're similar communities based on, you know, other than the tunnel. So I think we're just scratching the surface.
Yeah, that makes sense. It seems sort of obvious up front, but it's a heavier lift than sometimes is obvious, coordinating among different disciplines and really putting together a structure that works for approaching that stuff. But yeah, it seems like exciting opportunities.
I agree.
So what other questions are there for Robyn at this point?